Chapter 20, the immune system. I almost cut this one out. But I figured people are going to have a lot of time on their hands. They're going to want to learn about their immune system right about now. So I left it in. So the immune system can be broken down into two broad categories. The innate, try it like that. The innate immune system and the adaptive immune system. So the innate immune system, the things in your innate immune system, sometimes they're called the non-specific. The non-specific elements of your immune system. Because they can respond to broad types of challenges to your immune system. Whereas the adaptive immune system lends specific immunity. So when you hear about antibodies and immunity to specific pathogens, you're talking about the adaptive immune system. The innate immune system can just broadly respond to things like bacteria, like large classes of bacteria, viruses in general, not specific viruses. But it can do that very quickly. So in general, your innate immune system can adapt very quickly, whereas your adaptive immune system needs time to adapt its response. So this usually takes a little bit longer. And there's a difficulty when talking about the immune system, as you know, when learning the immune system, because when I'm teaching something like this, or really anything in this class, I try to break it down into discrete, digestible chunks of information. But with the immune system, there's so much going on, and it's just this symphony of moving parts and cells that are interacting and making antibodies and activating each other, and there's all kinds of stuff going on here. So it's a lot of moving parts all working together, and it's hard to talk about one specific thing at once without bringing in lots of other elements of the immune system. So I'm going to do this a whole lot in this lecture. I'm going to say, we'll come back to this because I'll introduce an idea and talk about that idea within the context of what I'm talking about at that moment, and then later on come back and talk about things like antibodies and stuff. So we'll start with the innate immune system because that is probably the easiest way to do it. So when I talk about the innate immune system, there's basically two broad things that go into it. There are your surface barriers, and internal defenses. So when I say surface barriers, basically I mean your skin, right, the integument. It's a physical barrier between the outside world and you, right? So your skin and your mucous membranes. So that's your first line of defense. Your skin is actually pretty acidic. I like to play guitar, and I got to change my strings a whole lot because my skin is acidic, just like yours, and so that acidity will corrode the strings on the guitar, and so they wear out after a few weeks. And it's just because of the acidity of the fluid on the skin. So that does serve a purpose, right? Because that'll actually kill some bacteria, not all bacteria, but it can kill some bacteria. And there's also a class of proteins secreted by your skin and your mucous membranes called defensins. If you look at the first PowerPoint slide, just a couple definitions I didn't want to write. Defensins, these are broad spectrum antimicrobial peptides produced by the skin and mucous membranes. So these are peptides, chains of amino acids that can target and kill broad spectrums of microbes. You've also got mucous membranes contributing to your surface barriers. Again, they're producing defensins. They make a lot of lysozymes, right? These are enzymes that can lyse bacteria. It's also acidic. And they produce mucin, which is where the name mucous membranes come from, because the surface is covered with that mucus, which is also on that first PowerPoint slide. It's just a protein that forms this thick substance when it dissolves in water. So that can trap pathogens entering the digestive and respiratory passages. So, that's what your boogers are. Your boogers are mucin, dried mucin that's trapped all kinds of dirt and pathogens that was going into your nose. The internal defenses are many and varied, and they're a lot more complex. So we'll start with a few that ought to be familiar to you. Most of you did well on the blood chapter. Phagocytes. Phagocytic leukocytes. So things like neutrophils and macrophages. These are phagocytic cells, and so the neutrophils are just kind of the first on the scene when there's a bacterial infection. Macrophages get their name. Macro, big eaters. They're big eaters. They eat lots of bacteria. So I think the next slide on the PowerPoint, I think it's a video. No, that's a picture of macrophages. So the macrophages reach out these long cytoplasmic arms and can grab onto bacteria and pull them in and then destroy the bacteria. Both macrophages and neutrophils track bacteria using a process called chemotaxis. So this is just how they can follow a trail of molecules left by a bacteria that they're following after. And this was actually the cellular mechanism behind this. I learned this in graduate school, and it was like the first thing that just kind of blew my mind, the way that this works. So it's a really cool thing. If you want to look into how the cytoskeleton actually polymerizes and depolymerizes in response to these chemicals, it's pretty neat. You should check it out. But you should at least watch that video, that YouTube video, because it's going to show you a neutrophil basically chasing after a bacteria. You can watch bacteria move around, and then the neutrophil just following right behind it until it latches onto the bacteria and smothers it and kills it. So that kind of brings us to our next topic, which is there we go. How phagocytic cells recognize and bind to their target pathogens. So the phagocytes need to be able to stick to the pathogen that they're killing. So there are a number of ways they can do this. One is via a class of cell surface receptors called pattern recognition receptors. So these are just receptors on the cell surface of various immune system cells that can recognize molecular patterns that are common among pathogens. So, I mean, we talked about the glycocalyx back in 2120, and even at the beginning of this chapter, talked about the glycocalyx. And I mentioned that bacteria can have a unique bacterial-like glycocalyx. So certain pattern recognition receptors allow cells of your immune system to recognize and bind to those bacterial glycocalyces. And so anything with a bacterial glycocalyx can get targeted by these cells that have the right pattern recognition receptors. Some bacteria get around this by producing a cell wall. So if you've already taken Med Micro, then you probably know what I'm talking about. If you haven't taken Med Micro, I'll just give you a taste of it. Some bacteria have a cell wall on the outside of their plasma membranes. And so what this will effectively do is it covers up their glycocalyx. So their glycocalyx is no longer exposed. So then the pattern recognition receptors can't recognize the glycocalyx. And so one way that our immune system counters that is through a class of molecules called opsonins. It's in the second definition on that slide. And it's basically just a molecule that'll bind to the pathogen, and then the cells of your immune system can bind to that opsonin molecule. And it's actually, it literally means, opsonization, like literally means to make tasty, right? So it's a way of sticking a molecule on a bacteria that can easily be recognized by one of the cells of your immune system, and so one of those phagocytic cells can then eat the bacteria or whatever pathogen that was targeted by that opsonin. So one really good example of an opsonin is an antibody, right? You've heard all about antibodies, and we'll go into painful detail about them later on in this chapter. But an antibody is an example of an opsonin molecule because it can bind to a pathogen, and then other cells can bind to that antibody and use that as a means to grab onto the bacteria or other pathogen and deal with it. Complement proteins, this is another example of an opsonin. We'll talk about complement here in just a minute. We'll talk about lectins in a minute. First, I want to talk about the complement system. We'll talk about complement now. Before we go into what actually happens when complement is activated, we'll talk about the three different ways that complement can be activated. One is the classical pathway. Basically, what happens in the classical pathway of complement activation is that an antibody will bind to a pathogen, and that activates the complement system. So an antibody binds to a pathogen, and that activates complement. The second way the complement system can get activated is via lectins. So there's a lectin pathway. And if you want to know what lectins are, look at that slide, the third definition on that first slide. They're water-soluble proteins that can bind to specific sugars on a pathogen surface. And when those proteins bind to the sugars on the surface of the pathogen, then that will activate the complement system. And the third way that the complement system can be activated is called the alternative pathway. And the alternative pathway is what happens when a cell doesn't have specific proteins that inhibit the complement system. This all makes sense to you in just a minute. Bear with me. So the alternative pathway, we'll call it spontaneous activation. Of the complement system. If the pathogens lack complement inhibitors. So before we go into what exactly the complement system is, right, I'll give you a broad overview. It's a way for your immune system to target and kill a really large variety of pathogens. And we'll go into the nuts and bolts of it here in a minute, but just bear that in mind right now, that it's a way for your immune system to target and kill a broad variety of pathogens, and it can be activated by antibodies, it can be activated by lectins, and it can also just spontaneously get activated via the alternative pathway if the pathogens are not expressing complement inhibitors. So a lot of the cells of your body express proteins that specifically inhibit the complement system. And so that's their way of just protecting themselves against this complement system that can really target and kill any kind of cell. So, how does the complement system do that? How does a complement system kill cells? Let's look at that. You got these three different pathways that can activate it, all right? Say the classical pathway, you've got the lectin pathway, and then you've got the alternative pathway. All these different pathways can activate the complement system. And what they'll do specifically is they will activate a protein called C3, right? This is a protein. C for complement, complement protein number 3. And when C3 gets activated, it will split apart into two different proteins, C3b and C3a. C3a goes on to promote a process called inflammation. All right, more on that later. But C3b actually directly contributes to killing whatever cell is being targeted by this complement. C3b is itself an opsonin. All right, that's one thing it does. C3b is an opsonin, so C3b can attach itself to the pathogen, and then other cells of the immune system can latch onto C3b and use that as a foothold to kind of grab onto the pathogen and kill it. Another thing that it does, all right, the other thing that it does is it'll combine with a number of other complement proteins So it'll combine with other complement proteins to produce this big multi-protein complex called the membrane attack complex. And we'll draw it like that. I'll show you what it looks like in just a second. But it's called the membrane attack complex, or MAC. Right? And so what it'll do is it'll actually, it's like a really thick channel protein that can insert itself into the membrane of a pathogen and allow water to diffuse in and out. And so when it inserts itself into the membrane of, let's say, a bacteria, right, there's MAC. When the membrane attack complex inserts itself into the membrane of a bacteria, then that allows water to get in, and so the cell will swell up and then lyse, and so it kills the bacteria. And I think on the next slide I've got a picture of the membrane attack complex. Yeah, so if you look at that next slide on the left, you can see the structure of the membrane attack complex, and then to the right, what you can see is a cell surface that has a whole bunch of MACs stuck in it. So it creates a whole bunch of pores in the membrane, so it lets a whole lot of water in. Right? Water is just going to rush into that cell and it's going to swell up and burst. So this can happen to a lot of different types of pathogens. It can happen to bacteria, and it can also be activated against cells of your body that are, either cancerous or infected with a virus. It's not just bacteria that get targeted by the complement system. All right. And you'll see later on that, or I think you might have noticed this already, this idea of the innate immune system and the adaptive immune system, these are not two totally discrete things, right? The complement system, we're talking about it right now in the context of the innate immune system. But you might have noticed, well, the complement system can be activated by antibodies. So the complement system, even though it's technically part of the innate immune system, it can still be activated by the adaptive immune system. So don't think that these are two totally discrete divisions of the immune system. There's a lot of overlap. Okay, so I told you we'd come back to inflammation. We'll talk about inflammation now. Inflammation is just your body's response to a number of different traumas. Physical trauma, heat, irritating chemicals, bacteria. These can all cause inflammation. So it's your body's response to physical trauma, excessive heat, irritating chemicals. Right? If you spill some kind of irritating chemical on your skin and it turns red and puffy, right? That's inflammation. It can also happen in response to viruses, fungi, bacteria, right? So broadly, the things that can cause inflammation can be lumped into one of two categories. Oops, try that again. Physical damage and pathogens. And it's basically just think of it as a quick way of mobilizing the innate immune system before the adaptive immune system can respond. So we'll go into what actually happens during an inflammation in just a minute. But first we'll talk about the things that can activate it and how they get activated, right? So the chemicals, the signaling molecules that can activate it are called cytokines. I'll try and spell that clearly. Cytokines, all right? These are the chemicals that are released by the cells of your body that can activate inflammation. One big one. Definitely heard of it. Histamine. You've probably heard of it in the context of antihistamines. So if you're like me and you get really nasty seasonal allergies, you can counteract your allergies by taking antihistamines. And basically, like all the symptoms of your allergies, the puffy eyes, the swollen mucous membranes in your nose and your respiratory tract, that's all inflammation, right? And a lot of it is activated by this chemical histamine. So people take antihistamines to counteract the inflammation that occurs in response to the things that they're allergic to. So histamine comes from basophils, another type of leukocyte we haven't talked about yet. Mast cells, we'll get to them later. Complement system can activate an inflammation. Remember when C3 breaks apart into C3A and C3B? C3A activates inflammation. And there are many other cytokines that can activate the inflammatory pathways. So what exactly happens during inflammation, right? All of this is going to make sense when you look at, the symptoms of inflammation. If you imagine an inflamed tissue, what do you imagine? You imagine something that's swollen and tender and red. And part of that, right, we're looking at the effects, part of that is because the arterioles in the tissue will dilate. And so you get a big increase in blood flow. So when an inflamed tissue swells up, part of that is because of the increased blood flow to the area, and it gets red also for the same reason. Well, we'll say 1, and then we'll say 2 is an increase in capillary permeability. So the capillaries will become a little bit more porous. And so what that does is among other things, it causes a release of clotting factors. So what you end up with is an interstitial clot. So a clot forms not inside the blood vessel, but in the surrounding tissues. And what that does is it kind of creates a temporary scaffolding for the repair of that tissue. Because in a lot of cases, one of the things that's causing the inflammation is something that can damage the tissue. You know, if you burn your hand on a hot stove, that's going to cause inflammation, but it's also going to kill a lot of tissue. And so that tissue is going to have to be repaired. So one of the things that that inflammation does is it causes the release of all these clotting factors, and a clot actually forms in the damaged tissue to just kind of temporarily hold everything together while, you know, so it makes time for the tissue to actually be repaired. And the third effect of inflammation is that it attracts leukocytes. So, we'll look at how that happens. Let's say, here's a blood vessel. Make it in red. That'll work. Here's a blood vessel, and here's a leukocyte. We'll make it a neutrophil. There we go. There's our neutrophil. The first step is that the neutrophil, or whatever leukocyte's being recruited here, it'll kind of attach itself to the edge of this capillary, kind of like this. So it will stick to the margin of the capillary. That's why this step right here is called margination. So margination is just when the leukocyte kind of sticks to the margin of the capillary. And then the leukocyte can just step out of the capillary. We call that diapedesis. And so now it's outside the capillary. Right? And it can just follow the trail of chemicals to the site of inflammation. And it does that by chemotaxis. Right? Remember I showed you that video, or you watched the video of the leukocyte following the trail of the bacteria, right? That's what it does. It'll just follow the trail of either bacterial chemicals, or it'll follow the trail of cytokines. And eventually it'll make its way to the site of inflammation. So all this stuff works together to promote healing. All these different elements of inflammation work together to promote healing of damaged tissue. Talk about another aspect of your innate immune system. All right, this one's kind of relevant. Interferons. These are molecules that are secreted by cells that have been infected with a bacteria. And what they do is these chemicals alert the surrounding cells that there is a viral infection nearby, and those cells need to get ready to deal with it. So here's how that works. Let's say here's a cell just hanging out, minding its own dam, and a virus comes along. So a virus will enter that cell, right? And it will start replicating. Once it starts doing that, there are mechanisms within this cell that allow the cell to release interferons. So the cell may have interferons just, inside a vesicle here within the cell. And if the cell gets attacked by a virus, then that vesicle can fuse with the plasma membrane and release the interferons outside the cell. So these are interferons right here. Interferons, IFN. All right. So these interferons aren't going to help this cell. This guy is doomed. It's too late for him. But maybe there's a neighboring cell that hasn't yet been attacked by that virus. And this cell has interferon receptors. So these interferons, which are released by the cell that was infected with the virus, can travel over here, bind to the interferon receptor, and what that'll do is it's basically this cell's way of telling that cell that I've been attacked by a virus and it's going to happen to you too. So get ready. So when this cell right here gets that message in the form of these paracrine signaling molecules called interferons, it will respond by expressing antiviral proteins. So what those antiviral proteins will do is inhibit that virus from entering this cell. So it's just a way of protecting itself from the virus. And so there's two things that it can inhibit. One is interferons can inhibit the entry of the virus, and they can inhibit the replication of the virus once it's in. Right? So just to recap, interferons are a way that a virus-infected cell can effectively warn its neighboring cells of a virus threat, and so that allows the neighboring cells to produce antiviral proteins that will slow down the spread of that virus. All right, another aspect of your innate immune system. Fever. All right, certain pathogens can cause macrophages and other leukocytes to release molecules called pyrogens. So these macrophages and other leukocytes will release pyrogens in response to detecting those pathogens. So the pyrogens wind up in the bloodstream, and they can make their way to the hypothalamus, which you may remember the hypothalamus is it's among a lot of other things. It's kind of like the central thermostat for your body. So the pyrogens cause the hypothalamus to basically raise your body temperature. And so this is a systemic response. And when I say systemic, I mean it happens throughout your entire body. You may only have a bacterial infection in your lungs, hopefully not, maybe just in the skin somewhere. But if it's enough to cause a fever, then that fever is going to be throughout your entire body. So that's what I mean by systemic. And basically, there's two benefits to this. One is it raises your metabolic rate. So it allows the cells of your immune system to work a lot faster. If they're hotter, right, they can their metabolic rate shoots up. They can get a lot more done with that high metabolic rate. And it allows your body to repair faster. I mean, all the cells in your body are just going to be working a lot faster if you got a fever. All right, and that's it for the innate immune system, and we're gonna, so we're gonna jump ahead to the adaptive. So on to the adaptive immune system. And the core relationship in the adaptive immune system is this relationship between antigens and antibodies. So we kind of skirted around this for, the better part of the year. So now we'll really get down to what exactly is an antigen and what is an antibody. So in a very broad sense, an antigen is just a substance that can mobilize an adaptive immune response. That's on your next PowerPoint slide, the one with antigen at the top. So an antigen is just a substance that can mobilize an adaptive immune response. These can be proteins or small peptides, right? Those really make the best, most effective antigens, but antigens can also be lipids, carbohydrates, nucleic acids. Pretty much any biomolecule can make a pretty good antigen. Any largish biomolecule can be an antigen. And the primary molecule responsible for recognizing antigens is antibodies. And I'll abbreviate them A, B, capital A, lowercase b from now on, so I don't have to write that over and over again. Antibodies are made by B lymphocytes. So we talked about B cells and T cells when we were talking about blood cells. But we're going to go into a lot of detail about how exactly B cells and T cells do exactly what they do. But first let's talk about antibodies. They're made by B cells, and they all have the same basic structure. They're proteins to start off. And so, you know, you understand a protein is just a chain of amino acids. Or a protein can be multiple chains of amino acids. A peptide chain is just a chain of amino acids, right? An antibody actually has 4 chains of amino acids. They have, let's see, we'll do black and red. 2 heavy chains and 2 light chains. So there are 4 peptide chains that go into an antibody. And they arrange themselves like this. You'll have a heavy chain here, and then another heavy chain, kind of like this. And then the 2 light chains arrange themselves like this. So you can see the heavy chain is longer, right? It's bigger, that's why it's called the heavy chain, and the light chain is much smaller. But any antibody is going to have, like, this is the core structure of an antibody. And they're symmetrical, right? So if we want to look at how they work, we can just look at one half. We can look at this half right here. All right? So we're going to look at this half of the antibody right there and straighten it out so it's a little bit easier to look at. So here's our heavy chain, and there's our light chain, right? All we did was we took this one, we straightened it out over here so we can look at this unit of the antibody right here. There are 2 regions, right? There is a variable region up here, and then there is a constant region down here. So these are called variable and constant regions because the constant region doesn't ever change. The thing about antibodies is that they can bind to specific antigens, right? This is, you know, the core, this is really what's at the heart of the adaptive immune system, because your adaptive immune system can detect and respond to very specific pathogens and very specific antigens, because antibodies can recognize very specific antigens. And it's because of this variable region, right? The variable region can be manipulated and rearranged so that it can respond to very specific molecules, whereas the constant region is always going to look the same. So if you take just a random antibody out of somebody's blood and look at the constant region, it'll be the same across all the antibodies. But what will be different is this variable region. So variations in the variable region are what allow the antibody to target and bind to specific antigens. If you look at the next slide, what you're looking at is just a typical antigen, or typical antibody. Immunoglobulin G, IgG. This is just a really common type of antibody. And what you can see is it's got the same basic structure that we drew here on the board, right? It's kind of Y-shaped. If you look at the picture on the right, you can really tell that there's 2 heavy chains and 2 light chains. In the picture on the right on that slide, there's 1 heavy chain that is yellow, and then another heavy chain that's light blue. And then that light blue heavy chain is bound to a dark blue light chain, and the yellow heavy chain is bound to a green light chain. Yeah. So the process by which B cells change the structure of their antibodies is really pretty neat. We're not going to go into the details of it because it's pretty complicated. But the reality is that there's only 1 gene that encodes a heavy chain, and there's only 1 gene that encodes a light chain. But there are millions of different combinations of the variable region, right? So what actually happens in a B cell is the variable region of that heavy and light chain gene gets rearranged within the genome. And the end result is that for any one B cell, any one active B cell that's making antibodies, it can only make 1 clone of that antibody. So for any one B cell, it can only make 1 antibody clone, right? So if you have a B cell that's making an antibody to, I don't know, let's say COVID-19, right? If you've got a B cell in your body that's making antibodies to that specific virus, that's the only kind of antibody that it makes. And there's a process that the B cells go through to specify their antibody specificity. And it's a really neat topic, and I wish I could do it justice. If you want to know more, take an immunology class, because it's pretty cool. Let's see. All right, so that was IgG. IgG is just one of several types of antibodies. In the next slide, what you're looking at is the 5 different classes of antibody. Immunoglobulin, antibody, just think of them as being synonymous for the purpose of this course. It's close enough. So these are the different classes of antibodies: IgA, IgD, IgE, IgG, and IgM. And they all have the same basic structure. IgG, IgE, and IgD all have that same 2 heavy chain, 2 light chain structure that I drew on the board. But they each elicit different responses from your immune system. So for example, IgD, if IgD binds to its antigen, then it will result in the activation of basophils and mast cells. So IgD is pretty heavily involved in things like allergic reactions, right? IgE, on the other hand, can protect against parasitic worms. So it'll also activate some allergic reactions, but when IgE binds to its antigen on a parasitic worm, that'll help eosinophils come in and bind to that worm. So I'm not going to go over each one, but I do want you to know, just in a very general sense, what type of response each IgG class can elicit. And I want you to note that they don't all have the exact same structure. You look at IgM, and it actually has 5 of those heavy chain, light chain combinations, all bound at their constant region, by, a scaffolding that holds them all together. So that allows IgM to bind to a whole bunch of different antigens. And this is why IgM is responsible for that blood transfusion reaction, right? When you get a transfusion that's not compatible with your blood type, IgM is the antibody that is responsible for that, because IgM will bind to multiple red blood cells and form this big insoluble lattice work of red blood cells and IgM antibodies, and it just forms that big, not even a clot, just like a big insoluble mess of red blood cells. That's the agglutination reaction that you were looking at a few weeks ago. Yeah, and that's actually, I didn't realize that's the next topic on my notes, is agglutination. All right, so this is what happens when the antibodies and the antigens form this big cross-linked lattice that is insoluble. So what I'm talking about here is I'm going to give you a number of different things that antibodies can do. Right? That's what we're talking about now. It's different things that antibodies can do. One trick is called agglutination. So the antibodies and the antigens form this big cross-linked lattice, and the cells will clump up. That's what we call agglutination. And this is what's responsible for transfusion reactions. And I got a picture of it on the next slide. There you go. Well, it's not really not a very good picture. I'd call that a cartoon, right? But you can see how the erythrocytes, right, the red blood cells, get caught up in this big, complicated network of erythrocytes and IgM. And you can see how, if you've got enough IgM antibodies, then it can create a really big complex. Another trick that antibodies can do, we'll call it neutralization. So basically, it's just what happens when the antibody binds to the antigen, and if the antigen has the antibody bound to it, then the antigen can't cause the problems that it was causing to begin with, right? So the antibody might block an important part of that antigen, right? So let's say the antigen may be a cell surface protein on a bacteria or a protein on a virus shell that allows that virus to stick to the outside of a cell and then make its way inside. It may be that the antibody can bind to that receptor on the virus, and now the protein on the virus, that important part of the protein, is blocked, and so the virus can't get into the cell anymore. Right? That's just one example. But it all starts with the antigen or the antibody binding to the antigen. And as a result, the antibody blocks an important site on the antigen. And now the antigen can't do what it was doing. And also, the antibody binding to the antigen targets that antigen for destruction by things like phagocytes. So the phagocytes will destroy the antibody antigen complex. Another cool antibody trick. Precipitation. This is just what happens when the antibody antigen complex precipitates out of solution. Right? That's what precipitate means. It means that whatever that thing is no longer in solutions. Like when we think of precipitation in the context of rain, right? When it's precipitating outside, what's happening is the water in the atmosphere just falls out of solution and condenses and falls down to earth and is liquid, right? So the same thing can happen to solutes that are in solution. They can just kind of stick together and form a solid and fall out of solution. So this is what you get when a soluble antigen gets an antibody stuck to it, and the result is an insoluble complex that just falls out of solution, and then phagocytes clean up the mess. Because the phagocytes can recognize the antibodies pretty easily. They can recognize the constant region of the antibodies pretty easily. And the last trick that antibodies can do, and arguably one of the most important, is complement activation. So when an antibody binds to a pathogen, then that can activate, if the pathogen is a cell, then that can activate complement. You already know what the complement system does. It results in lysis of the invading cell. So, if we were to summarize all the things that antibodies can do, the antibody functions, they can cause precipitation, they can cause lysis via the complement system, they can cause agglutination, right, just like in a transfusion reaction, and they can cause neutralization. It's not up here. Already did it. Precipitation, lysis, agglutination, and neutralization. There you go. There's your mnemonic right there. Plan. It's a good way to remember this stuff. I give you one more definition, and then I'm going to take a break because I've been talking for a minute. There's your definition, right, on the next slide. Epitope. The epitope is just the part of the antigen that is targeted by an antibody. So if you have an antibody that targets a big protein, there may only be one small part of that protein that's actually recognized by the antibody. And that one small part of the protein that's recognized by the antigen, the antibody, is called the epitope. All right, so now we're going to talk about B-cell activation. And before we jump into this, I just want to talk a little bit about the need for B-cell activation. So think of B cells as, their only job is to make antibodies, right? That's all they do. They just make antibodies. And And it wouldn't be very helpful if every B cell in your body was making antibodies all the time, right? Because every B cell makes a specific antibody that can target, ideally a specific antigen. But there's no reason to make the antibodies if the antigen isn't present. So there's a process that they go through called activation that basically switches on a B cell, and it basically allows the B cell to ramp up its antibody production in response to encountering its antigen. So keep that in mind when we talk about B cell activation. It's just the process by which B cells get switched on and effectively get told to make their antibodies. So it all starts with what we'll call a naive B cell. Naive, right, means the same thing in the context of B cells as it does in the context of, a person being naive. And if you say a person is naive, it means that they've never, they're not familiar with whatever you're talking about because they've just never encountered it, right? Like, somebody who maybe grew up in a really loving household where, there are no racists in the household. They may think, well, I grew up in a southern home where everybody was really nice to everybody, and there are no racists where I grew up, so I don't think racists exist. They're just naive, right? There's lots of racists, but that person is naive because they've never encountered them. A naive B cell is naive in the sense that it has never encountered its antigen. It makes antibodies, or it can make antibodies, but it's never encountered an antigen that just really sticks to that antibody very well. So a naive B cell will make antibodies. It won't make a lot of antibodies, but it will make some antibodies, and it will express those antibodies on the outside of its plasma membrane. So those antibodies will just be kind of stuck along the outside of the membrane of this naive B cell. Got a lot of colors here. Okay, so the first thing that happens in this process of activating a naive B cell is it has to encounter its antigen somehow. It has to encounter some kind of molecule that sticks to its antibody. We'll call that the antigen, and we'll draw it like this. Right, there's the antigen. It'll stick to this antibody. There you go. Once an antigen sticks to that antibody, the cell will endocytose that antibody-antigen complex. And so what you'll get is a vesicle with the antigen inside of it. There you go. So that's step 2. Endocytosis. And then the cell, the B cell, will take that antigen inside the vesicle and stick it on a protein called the major histocompatibility complex. And it will express that antigen on the major histocompatibility complex on the outside of the cell. So it will look like this. So there you go. So if that antigen binds to the antibody, the cell will bring in the antibody and the antigen into a vesicle. It'll take that antigen, stick it on a major histocompatibility complex, and express it on the outside of the cell. I'll write down that for you. The major histocompatibility complex number 2, or we'll just call it MHC2 for short. That is this thing right there. So before we jump ahead to what happens next, think of it this way. Think of it as a way for the B cell, before it goes on to make a whole bunch of antibodies, this B cell needs to verify that this is actually something bad, right? The B cell needs to verify that this is not just a protein that's made by some other cell in your body, right? There's a possibility, a low possibility that could happen, but there needs to be a way in place for this B cell to verify that the antigen is what we call non-self, right? It wasn't made by us, self, right? So MHC2 is just the B cell's way of essentially holding out the antigen and saying, I found this, right? This showed up and stuck to my antibody, and this is it. So it's basically holding out that antigen for another part of your immune system to come and check it and say, oh yeah, that's bad. We need to make a bunch of antibodies to fight that off. So the other cell that does that is called a helper T cell. So this right here is a naive B cell, but the cell that's going to come over and check to make sure that that antigen is something that's not supposed to be here is called a helper T cell. Or we'll just call it T subscript H, or helper T cell. Remember, T cells are the other kind of lymphocyte. We're talking about lymphocytes here. Okay, so the way that a helper T cell can verify whether or not this antigen is something bad or not is via the activity of its T cell receptor. So that's what I've got in orange right there. That is the T cell receptor, or TCR. Right? So the T cell receptor will bind to the MHC antigen complex, and another protein on the T cell will bind to just the major histocompatibility complex. This accessory receptor is called CD4. So the helper T cell has two ways of binding to this structure. One is the T cell receptor that binds to the antigen and MHC. And then the other is the CD4 receptor that reaches around and binds to the major histocompatibility complex. So we'll call all of this whole dance that they're doing right there, step number 4, right, is just the helper T cell binding to MHC and the antigen via the T cell receptor, and binding to MHC via the CD4 receptor. If the helper T cell identifies the antigen as non-self. I would put this in big fat stars. If the helper T cell identifies that antigen as non-self, then and only then will the helper T cell release interleukins, which are just paracrine signaling molecules. They get released and they can signal the B cell, right, to start dividing and making more antibodies. So that'll be 5. All right. But I want to reiterate again that this only happens if the T cell recognizes the antigen as non-self. And what I mean by non-self is the T cell will recognize that antigen as something that was not made by your body. Right? So it could be a viral protein, it could be a bacterial protein, it could just be, some piece of viral RNA that got picked up somewhere. Could be anything. It's whatever bound to that antibody. So the effect here is that, if this B cell just happens to pick up an antigen that, was made by your body somewhere, it'll pick up that antigen, endocytose it, hold it out on MHC2, and then a helper T cell will bind to it and basically just not do anything because the helper T cell will recognize that antigen as something that's, supposed to be there. So if that's the case, this doesn't happen, right? If that antigen is something from your body, then if this is all working correctly, the helper T cell won't release interleukins, and then the B cell won't get activated, right? But if the helper T cell recognizes that antigen as non-self, then it'll release the interleukins, and the B cell will differentiate. It'll divide and differentiate into 2 types of cells. Step 6. One is a memory B cell, and the other thing it can differentiate into is called a plasma cell. And a plasma cell is basically I can't talk, I'm right. A plasma cell is basically just an activated B cell that makes a whole hell of a lot of antibodies, right? And so that's the way this works. If a B cell, a naive B cell, picks up its antigen, presents it, and then gets stimulated by a helper T cell, by these interleukins, then it will respond by differentiating into these 2 types of cells. The plasma cell that makes a whole bunch of antibodies, so now we know that the body is being invaded by whatever kind of antigen, whatever this antigen came from. So it's going to produce a whole bunch of antibodies to deal with that pathogenic invasion. And it will produce memory B cells, which are basically just pre-activated B cells. All right? They're B cells that have already been activated. And all they need is just a challenge from the antigen that binds to their antibody. So if a B cell, if a memory B cell encounters its antigen, it's already been activated. It doesn't have to go through this dance right here. If a memory B cell encounters its antigen, then it'll just go straight into differentiating and making more plasma cells. So this is how B cells first encounter their antigen and then verify that their antigen is non-self and then make a whole bunch of antibodies. I'm going to erase this. So now the next time the antigen shows up, there are already a whole bunch of memory B cells already activated, already ready to go. If they encounter that antigen, they don't have to get any help from the helper T. They can just go straight to making plasma cells and straight to making antibodies. So that's why the first time your body encounters an antigen, you get a very small response in terms of antibody production. But the second time, you get a really big one. All right, so what we're going to graph here is time on the x-axis, and then on the y-axis, we're going to graph titer. And that's the next definition on that slide, the last slide that we looked at with epitope. Titer. Titer is just a fancy word for the concentration of an antibody. So, the first time you ever get exposed to an antigen, if you've got a B cell that can make antibodies that will target that antigen, then the B cell will engulf it and present it to a T cell. T cell will verify that it's non-self, and the T cell will release the interleukins, and the interleukins will cause that B cell to differentiate and make a few plasma cells and some memory B cells. And those plasma cells are going to make some antibodies, right? So you get a pretty slow, gradual increase in titer, increase in that concentration of that antibody. This is after the first exposure. This is the first exposure to a particular antigen. And we call this response right here a primary immune response. That. This gradual increase in titer, and then it falls off. Right? But even after that immune response tapers off, after the titer goes back down, you still got memory B cells, right? So, if you get a second exposure to that same antigen, you already have pre-activated B cells. You've already got memory B cells ready to go. All they need to do is encounter their antigen, and then they're going to switch on and make a whole bunch of plasma cells, make a whole bunch of antibodies. So the second time you get exposed to that antigen, you get a massive, very fast increase in antibody production. And this is orders of magnitude bigger, right? Way bigger and way faster. So this can be up to 1,000 times stronger. This can result in up to 1,000 times more antibodies produced than in the primary immune response. And we call this a secondary immune response. Right? It is up to 1,000 times stronger, and it's faster because this time, or the first time around, the B cell had to do the dance with the T cell. The B cell had to verify with the T cell that the antigen was something that we need to fight off, and then differentiate. It takes time. This time around, the second time around, that's already been done. T cells already verified that that's a non-self antigen, and so this time around, it happens much faster. And you already had a whole bunch of memory B cells, so you're able to make a lot more plasma cells in a much shorter amount of time. This is why we vaccinate, right? When you get a vaccination, and you may get a little inflammation. If you get a shot right there in your shoulder, if you get a flu shot, you may feel a little bit sore the next day right there, and that's because of the inflammation. And it may take like a day or two to run its course. That's this right here, right? But then, let's say a couple weeks later, you actually get exposed to the flu. All right, your B cells have already seen that antigen. So the B cells have already been activated, and if they encounter that flu antigen, they can ramp up antibody production real quick. And so your body can fight off that flu a whole lot faster if you just go ahead and pre-activate your B cells. So think of that. Think of vaccinations that way. Think of vaccinations as a way to pre-activate your B cells to an antigen that you're likely to encounter. All right, next slide is basically just showing you what's going on during the primary and the secondary immune response. I think that's straight out of your book. I think it's figure 20.11. Yeah. And the top part of that picture, it's just showing you what happens in the primary immune response, right? You may have multiples, on that top row, you may have multiple different B cells. Each one makes a different antibody, and then an antigen shows up. Only the B cell that recognizes that antibody, or only the B cell that recognizes that antigen can get activated by that antigen. So the clone, think of each B cell as being a clone of itself, right? The clone that responds to that antigen gets selected and expanded. So that one clone line gets expanded, and you end up with lots of B cells that are all clones of that original B cell that originally recognized the antigen. And the first time around, you get a few plasma cells, a few memory B cells, but then the second time that one of those memory B cells encounters an antigen, it can produce a whole lot more plasma cells and even more memory B cells. And these memory B cells can last a really long time. I mean, you're immune to things that you were exposed to decades ago, if you've been alive for decades. Well, you've all been alive for decades. But those memory B cells can stick around for a long time. All right, the type of immunity that is imparted by B cells is called humoral immunity, right? This is the type of immunity imparted by B cells, because when B cells make antibodies, they just spew antibodies out. They just make antibodies and then secrete them. They're just exocytosing them and releasing those soluble antibodies into the humor of the body. They're releasing antibodies into the fluid of your body, right, which we call the humor. That's why it's called humoral immunity, because they're just putting all those antibodies into your bodily fluids. B cells don't actively do anything against pathogens. They don't actively target pathogens other than just making antibodies. So a B cell may make IgG antibodies. And so an IgG antibody, once it's made by a B cell, it goes off into the humor of the body. It goes off into the bodily fluids. And if it encounters its antigen, it'll stick to the antigen. If that antigen's a bacteria, then the antibody sticks to that bacteria, and then another cell, the phagocyte, can come along and use that opsonization as a means to target the bacteria. But B cells don't directly interact with the bacteria in terms of fighting it off. Talk about one more thing antibodies can do. Your integument and your mucosal membranes are filled with cells called mast cells. And basically, mast cells are cells that are filled with vesicles containing histamine. We'll make it red. These are vesicles filled with histamine. And these mast cells, they can just pick up antibodies, right? Like I said, the B cells are just making antibodies and releasing them out into the humor of the body. One of those antibodies, let's say IgE, right? Those antibodies can insert themselves into, I don't know why I did that. They can insert themselves into the plasma membrane of a mast cell. So this mast cell can have antibodies from lots of different B cells. So it can respond to a whole bunch of different antigens, because each B cell is going to make antibodies that can target a different antigen. So it's covered in antibodies. And let's say an antigen shows up that binds to one of those antibodies. All right, we'll do our antigen like that again. If that antigen binds to one of the antibodies on the surface of this mast cell, then all these vesicles will fuse with the plasma membrane. A process we call degranulation, because these vesicles sometimes are called granules. So let's say that. Granules or vesicles filled with histamine. So when an antigen binds to an antibody on that mast cell, then the mast cell will degranulate. And so when a mast cell degranulates, I'll spell that for you. Degranulation. When a mast cell degranulates, it's just spewing histamine everywhere. And you know what histamine does now, right? Histamine is a very powerful inflammatory cytokine. So this can cause a lot of inflammation. And you want to see what it looks like. The next slide there, labeled degranulating mast cell. On the left is a mast cell before degranulation. And then on the right is what happens if that mast cell encounters its antigen. So yeah, there's a lot of these in the mucous membranes of your body and a lot of these in your skin. If you have allergies, that guy is what's responsible for most of why you feel like crap when you have your allergies. It's because it's going to release a lot of histamine. When a mast cell does that in response to an allergen, right? An allergen is just something that causes allergies. That's an example of what we call an acute or a type 1 hypersensitivity. Type 1 hypersensitivity. In this case, I mean, hypersensitivity is just when your immune system is overly sensitive to something that's not necessarily damaging, right? So in this case, the antigen is what we call an allergen, right? I have allergies. I don't know what I'm allergic to. I have seasonal winter allergies. A lot of people are allergic to ragweed pollen. That's a really common one around this time of year. Ragweed pollen is not going to kill you. If your immune system didn't freak out about it, then you wouldn't have any idea it was there. So this is why allergic reactions are so pesky, because they're totally unnecessary. So an acute or type 1 hypersensitivity results in what we call an allergic reaction. Which is basically just inflammation. Right? The mucous membranes in your nose swell up, and you can't breathe through your nose, and then your eyes swell up, and you start, making a lot of tears, and your eyes get red, and it's all just inflammation. And those kinds of allergies are annoying, right? But they're usually not deadly unless the allergen gets into your blood. If the allergen gets into your blood, then that can cause something called anaphylactic shock, right? This can be a problem. This is what you get when you see systemic systemic allergen exposure. So, you look at that mast cell, and just looking at that thing makes me want to sneeze. Imagine if the allergen got into your bloodstream, and instead of just having that allergic reaction on your mucous membranes, instead that allergic reaction happened throughout your entire body, right? So instead of just, getting a puffy, red, inflamed face, you get a puffy, inflamed, red body, right? So one of the things that happens during inflammation is the blood vessels dilate quite a bit. So if that happens systemically, then you can get a huge drop in blood pressure. So if you get a systemic allergen exposure, then what you get is systemic inflammation. And so that can be very life-threatening. People that are allergic to bee stings, that's like the classic anaphylactic shock example. Because, I get stung by a bee, I don't care. It doesn't feel good, but it's not going to kill me because I'm not allergic to them. But if you're allergic to bee stings, when you get stung by a bee, that bee is basically injecting the allergen into your blood. And so the allergen goes throughout your entire body, and you get this systemic inflammation. And so that's why people that have those types of allergies, right? People that are allergic to bee stings, they carry epinephrine pens with them all the time. because that's the best way to treat it. The thing that kills you during anaphylactic shock is the drop of blood pressure. So that's why epinephrine can counteract it, right? It's just adrenaline. It's going to jack your blood pressure back up so that that drop in blood pressure doesn't kill you. And it'll also open up your airways. That's another thing that'll happen. You know, not only do your blood vessels dilate, but the airways will constrict. Right? If they get inflamed, then they'll constrict and then that person can't breathe anymore. So epinephrine will counter both of those things. So it'll open your airways and reconstrict the blood vessels. So that's type 1 hypersensitivity. Next one is called a cytotoxic or type 2 hypersensitivity. This is a type 2 hypersensitivity, cytotoxic. This is what you get if the antigen is on a cell. An example here is a transfusion reaction, right? What happens to red blood cells during a transfusion reaction? That's a type 2 hypersensitivity. If the antigen is widespread and the antigen and antibody form these large complexes, then what you're looking at is an immune complex or type 3 hypersensitivity. So here the antigen is widespread. And this is a type 3 if the antigen and antibody form these insoluble complexes. And what you get as a result of that, you know, these complexes will fall out of solution, right? They'll precipitate out of solution, and you get an intense inflammatory response. An example here is lupus. If you know anybody who has lupus, that's an autoimmune disorder where the antigen, when it binds to the antibody, they just precipitate out of solution, and it can cause these very severe hypersensitivity reactions. They're a little bit slower than a type 1. So in general, as you go from type 1, type 2, type 3, you're looking at slower and slower reaction time. So an acute hypersensitivity can happen in a matter of minutes. But type 3 hypersensitivity can last for several hours. And then a type 4 is the slowest one. So type 4 hypersensitivities are called delayed hypersensitivities. These can take 1 or 3 days to appear. And this is one, oops, this is a hypersensitivity that's actually mediated by T cells. And this just results in local inflammation. And an example, a classic example of this is contact dermatitis. Right? What happens when you get poison ivy? Take a break. I've been doing this too long. Okay, yeah, so when you get poison ivy, if you go outside and you wade through a bunch of poison ivy and you don't wash your legs or your feet or whatever, it might take a day. It might take a day for that poison ivy to show up. So, yeah, that's why these are called delayed hypersensitivities. Nickel, that's another. If you got a nickel allergy, that's another example of a delayed hypersensitivity or contact dermatitis. Okay, I'm going to take a break and then I'll come back and talk about T cells. All right, T cells. On to T cells. The other lymphocyte. T cells are called T cells because they mature in the thymus. T for thymus. If you look at the next slide on the PowerPoint, you'll see 2 examples of a thymus. On the left one in an adult and on the right one in an infant. And what you'll notice is that the thymus is much larger in an infant because that's when T cells are going through their process of clonal selection, which we'll get into in a little bit. But yeah, so I haven't really said this yet, but B cells are called B cells because they mature in the bone marrow, and T cells are called T cells because they mature in the thymus. So T for thymus, B for bone marrow. So those are T cells. Their surface is covered in T cell receptors. And the T cell receptors kind of function like antibodies. We'll talk about them in just a minute. But I do want to stress that T cells don't make antibodies. Right? Antibodies are exclusively made by B cells. T cells make T cell receptors. And that T cell receptor is very similar to an antibody in that it's what the T cell uses to target whatever antigen it can recognize. So let's talk about those T cell receptors. These are a cell surface only. All right, that's one difference between them and antibodies. Antibodies are made by B cells and then just released into the humor of the body. T cell receptors are only expressed on the surface of T cells. One thing they have in common with antibodies is they have a constant and a variable region, just like an antibody. So structurally, they're very similar. But one thing that really sets them apart from antibodies is the fact that a T cell receptor, I put a star next here, because this is a big one. T cell receptors can only recognize their antibody if that antibody is presented on a major histocompatibility complex. So they only recognize the antigen if that antigen is presented on the major histocompatibility complex, right? Either MHC1 or MHC2. We've only talked about MHC2 so far, right? MHC2, that was on the B cell that we were talking about that was activated. But there's also an MHC1, which we'll talk about in just a minute. Look at the next slide. And what you can see is just the general structure of a T cell receptor, right? They're a little bit, they're different from antibodies in that they only have two chains, right? There's an alpha chain and a beta chain. They don't have two heavy chains and two light chains. They just have an alpha chain and a beta chain. But they do have that same constant and variable region. So the constant region on a T cell receptor is never going to change, right? Because that's the region that the T cell receptor embeds in the plasma membrane of the T cell. But the variable region, just like the variable region on an antibody, can change quite a bit, right? Different, or very early on during T cell development, the gene that encodes that variable region for the alpha and the beta chain, it'll rearrange. And so every T cell that comes out of the thymus has a completely random variable region that allows it to attach to different antigens. So T cells are classified, there's two varieties of T cells. And these T cells are categorized based on which accessory receptor they express. So if you remember back when we were talking about B cell activation, go back and look at your B cell activation notes. Hit pause while you find your B cell activation notes. And now that you found them, look at the interface between the helper T cell and the B cell. And you'll notice that there's a T cell receptor that binds to the MHC and antigen complex. But then there's also that accessory receptor CD4 that's embedded in the helper T cell membrane, and it binds to the major histocompatibility complex, right? So that CD4, that's the accessory receptor that we're talking about here. So there's two varieties of T cells. One that expresses CD4, we call these CD4 positive T cells. And then there's another one, there's another kind that expresses a different accessory receptor called CD8. So we call these CD8 positive T cells. Well, obviously the CD4 positive T cells have the CD4 receptor, and CD8 positive T cells have the CD8 receptor. CD4 positive T cells bind to the major histocompatibility complex number 2, and CD8 positive T cells bind to MHC1, or I should say over here, CD4 positive T cells bind MHC2. Here we go. All right, here's an easy way to keep this straight. Hopefully, well, I don't know, maybe you haven't read this book. There's a book called The Hitchhiker's Guide to the Galaxy. It's a really good book. You got a lot of time to read right now. You should read this book. It's by a guy named Douglas Adams. And in this book, there's a race of people in the distant future, and they're trying to figure out what the meaning of life is. What they want to know the answer to the ultimate question of life, the universe, and everything. So they build this gigantic computer, and they write this program that takes billions of years to run. And they write this computer program, and the computer program is designed to answer that ultimate question of life, the universe, and everything. And then they just, hit run, and they run the program, and it runs for billions of years. And then, their entire religion is built around this computer program that's running. And, as billions of years pass, and then finally the computer program finishes its execution. And it comes up with an answer, and then they throw this huge festival around finding out the answer to the question. And it turns out the answer to the question, the ultimate question of life, the universe, and everything, is 42. Right? That's the answer. And they realized they didn't know what the question was to begin with, so it has no meaning. Anyway, it's a good book. Funny book. All right. 42. MHC2 binds to CD4, or CD4 binds to MHC2. 42. It's the answer to the ultimate question of life, the universe, and everything. That's how I remember it, but only because I read the book. I don't know if I said the name of the book. This isn't on the test. The Hitchhiker's Guide to the Galaxy. Douglas Adams. All right, so those are the two accessory receptors. And so CD4 positive T cells can only bind to MHC2, and CD8 positive T cells can only bind to MHC1. So who cares, right? What the heck is MHC1 and MHC2? I've kind of alluded to it. We'll talk about MHC1 and MHC2. You already kind of know what the function of MHC2 is. So we'll start over here, right? Go back and review B-cell activation, right? You can even listen to when I was talking about B-cell activation on this very video. And you should get a pretty good idea of what the purpose of MHC2 is. It's a way for a B cell, when it engulfs its antigen, to hold out that antigen, and basically it's saying, I found this, right? My antibody stuck to this antigen, and I need to know whether or not we should make antibodies to it. And I hate anthropomorphizing cells, but I think that's a good analogy to think about what's going on here. So the cells that express MHC2, these are called antigen-presenting cells. Right? Antigen-presenting cells, or APCs for short. All right, so things like B cells, right? They're called antigen-presenting cells because they are literally presenting an antigen on MHC2, and it's an antigen that they picked up somewhere else. So B cells do this, macrophages can do this. All right? You know what macrophages do? These are big phagocytic cells. So when they engulf a bacteria, they'll break up that bacteria and take little pieces of it and stick it on MHC2 and hold it out and say, hey, we found this. Is this something that we need to mount an immune response to? Another type of cell we haven't talked about yet, dendritic cells. Right? These are other antigen-presenting cells. But basically, the thing they all have in common is that they're taking an exogenous antigen and presenting it on MHC2. And it's an exogenous antigen because it came from outside the cell. And so it's something that they phagocytized. It's something that they picked up, something they phagocytized from outside the cell and then presented it on MHC2. That's what gets put on MHC2. MHC1, on the other hand, is expressed by all nucleated cells. Every cell in your body that has a nucleus expresses MHC1. And what they do is, it functions basically the same way. It's still a protein complex that allows the cell to present various antigens. But whereas MHC2 is presenting exogenous antigens, stuff that came from outside the cell, MHC1 is presenting endogenous antigens. So what that means is, just think of any cell in your body. Pick your favorite cell that you've learned about this year, like neurons, right? Just think of a neuron. So it's got a nucleus and it's making a lot of proteins, right? Neurons make a lot of proteins. Some of that protein, all the different, thousands and thousands of proteins that are being made in that cell, most of them are used to do whatever that protein does, but some of them get chopped up and stuck on MHC2, right? So for maybe every 1,000 sodium potassium pumps that are made by that cell, maybe one of them gets chopped up, stuck on MHC1, and then presented on the outside of the cell. And so the proteins, the antigens that are being put on MHC1 came from within the cell. It was stuff that the cell was already making. There's one exception to this. One exception is dendritic cells. Right? We heard about dendritic cells over here. Dendritic cells can, take exogenous antigens and stick them on MHC2. But they can also take exogenous antigens and put them on MHC1. So dendritic cells can present dendritic cells can present exogenous antigens on both MHC2 and MHC1. So that makes them kind of unique. So with the exception of dendritic cells, MHC1 is basically a way for a cell to say, I made this. Right? Every protein that's being made by that cell, some of it will get chopped up and stuck on MHC1, and the cell will just hold that out on the outside of the cell as a way to tell the immune system, hey, this is a protein that I'm making. right? And so this comes into play when, let's say, the cell starts making proteins that it shouldn't make. In the case of, I don't know, a virus. So if a cell gets invaded by a virus, one of the things that viruses do is they hijack all the machinery that makes proteins in a cell, and so that cell will start making viral proteins. Well, if that cell makes a whole bunch of viral proteins, some of it's going to get chopped up and put on MHC1. And it's going to hold that MHC1 out with the antigen, with the viral protein on it, and it's basically saying, I made this. And so various cells of your immune system can recognize that. Well, actually T cells, not various cells. T cells can recognize that, and if the T cell can target that viral antibody, then the T cell will kill the cell that's expressing that viral protein. So, yeah, make sure you understand how MHC1 and MHC2 differ and what different functions they serve. So MHC1, again, I'll say it again. MHC1 is a way for a cell to basically show a T cell what kinds of proteins it's making, whereas MHC2 is a way for an antigen-presenting cell, some kind of phagocytic cell, to show what kind of antigen it encountered, right? What kind of antigen it engulfed, right? Not something that it made. Okay. So, let's talk about T cell activation. All right. So those antigen-presenting cells are pretty important when we're talking about T cell activation. I'll show you a picture of a dendritic cell. There you go. That next picture on the slide, on the PowerPoint, is of a dendritic cell. They kind of look like macrophages. They kind of act like macrophages in that they're phagocytic, right? If they encounter some kind of pathogen, they will engulf that pathogen and then express antigens from that pathogen on the outside of the cell. So we'll draw that. Let's say here's a dendritic cell. All right. There's a dendritic cell. And it's just hanging out, and we'll call it the peripheral tissue. What I mean is, it's not in a lymph node. It's, it's, I don't know, in the skin, or it's in a mucous membrane, or it's just somewhere in your body. It's just hanging out. And let's say it maybe encounters some kind of pathogen or some kind of antigen. Let's say a bacteria. It engulfs a bacteria and then presents an antigen from that bacteria on MHC. So it engulfs that antigen and then presents the antigen on both MHC1 and MHC2. Because remember, dendritic cells are the only kind of cell that can do that, right? So it will present this exogenous antigen on MHC1 and MHC2. So one there and a one there. So this is MHC1 and 2. So if that happens, right, this dendritic cell encounters an antigen, engulfs the antigen, presents it on MHC1 and MHC2, then it will hop into the lymphatic system. And it will travel to a lymph node. And so now here's our antigen presenting cell, our dendritic cell, in the lymph node. Right? This is a dendritic cell that's presenting antigen, so it's an antigen presenting cell. It's presenting an antigen on MHC2. And on MHC1. So this is MHC, I don't know if you can read that. This is MHC2. This is MHC1. Oops, I didn't finish drawing my antigen. There we go. All right. So here's our antigen presenting cell in a lymph node presenting the antigen on MHC2 and MHC1. Now, before we go any further, remember what the name of the game here is: activating T cells. And basically what's happening here is the T cells, a T cell can't engulf an antigen on its own. It can only recognize its antigen when that antigen is presented on MHC2 or MHC1, right? So the T cell can't recognize its antigen without some kind of help from another cell, like an antigen presenting cell. So first we're going to talk about CD4 positive T cell activation, right? This is one you're already a little bit familiar with CD4 because we looked at it when we were talking about B cell activation, right? So here's our CD4 positive T cell. It's naive, right? It's not active yet because it hasn't encountered its antigen yet. But it's got, I don't remember what color I did the main T cell receptor in. We'll go with black. So there's our T cell receptor. And there's our CD4 receptor. So this CD4 positive T cell will bind to MHC2 via the T cell receptor and the CD4 receptor. We'll call that step 1. And then if, and this is a big if the T cell receptor recognizes the antigen as non-self, then that CD4 positive T cell will differentiate into a helper T. And now that helper T can then go on to help B cells get activated. So before we go into the other kind, the CD8 positive T cell, just look at this as, a discrete thing that can happen that contributes to the overall immunity, right? Let's say you get, you encounter an antigen. You encounter an antigen that's foreign and it's some kind of pathogenic antigen. Let's say a virus, okay? So two things need to happen. Your immune system needs to start making antibodies to that virus. And yeah, so you need to start making antibodies to the virus. Well, what's going to make the antibodies? B cells. B cell is only going to make an antibody to that virus if the B cell encounters the virus and its antibody matches up with that viral antigen easily. So the B cell will engulf that virus, present the antigen on MHC2, and just wait for a T cell to show up, a helper T cell. It'll wait for a helper T cell to show up, bind to the MHC2 antigen complex and say, oh yeah, we need to kill that, right? So first the B cell has to encounter the antigen and present it on MHC2, and then we need a helper T cell to bind to the MHC antigen complex and verify that that protein or that antigen is non-self. But before a helper T cell can do that, the helper T cell has to be activated by an antigen presenting cell. So that's the other thing that has to happen, right? So an antigen presenting cell, right, a dendritic cell, has to find that antigen. So that antigen's got to get has to be found by not only a B cell but also a dendritic cell. So the dendritic cell will encounter that antigen. There's our viral antigen right there. Dendritic cell will bring it in, express it on MHC2, hop into a lymph node. Here it is in a lymph node with that antigen presented on MHC2, and then a helper T cell. If the helper T cell has a T cell receptor that recognizes that antigen, then the helper T cell can bind to the MHC2 antigen complex. It gets activated and it becomes a helper T cell, and now this helper T cell can go on to help a B cell get activated. That's why it's called a helper T cell. So what this does is it creates a number of roadblocks to creating an antibody. And that's a good thing, because you don't want a B cell to start making antibodies every time it encounters any old antigen that it binds to, because there's a good chance that, you know, a B cell can encounter a protein made by healthy cells that excites that B cell. And if the B cell starts making antibodies and pumping them out that target that normal protein, then you end up with an autoimmune disease. So these are just checks and balances in place that keep that from happening. Okay. Now, that is just CD4 positive T cell activation. I'm going to tell you how CD8 positive T cells are going to be activated, but I'm just going to go ahead and make a prediction that people are going to confuse this and think it's all one thing. All right, we're done talking about CD4 positive T cells. All right, so what I would do is I would just say this is helper T cell activation right here. All right, this is helper T cell activation. We're going to talk about CD8 positive T cell activation, but it's going to start the same way. So I would go to a new sheet in your notes if you're taking notes. I would go to a new sheet in your notes and I would just copy all of this down, right? Because we're just going to build on top of it for CD8 positive T cell. But what I want to stress here is that at this point, helper T cell activation is done, right? This is the process of helper T cell activation. So you pause the video, make sure that you basically created another sheet on your notes, because we're going to now talk about cytotoxic T cell activation. All right. So, cytotoxic T cell activation starts the same way. Let's say this time, let's say, you know, that virus that we were talking about, that virus will get into the cells of your body and it'll hijack the machinery and it'll make those cells start to produce viral proteins. And the viral proteins get expressed on the outside of the cell. Well, now your immune system needs to start killing any cell that's infected with the virus. That's how your body deals with viral infections. One of the ways. So we need to activate some T cells that will kill any cell expressing that viral protein. So that's what cytotoxic T cells do. And the way they get activated starts out the same way. All right, a dendritic cell will encounter the antigen, engulf the antigen. This is a viral antigen right here. It will engulf that antigen, express the antigen on MHC1 and MHC2. It will hop into a lymph node, or it will hop into the lymphatic system and travel to a lymph node. So here we are in a lymph node. And the antigen presenting cell has the antigen expressed on MHC1 and MHC2. And then a naive cytotoxic, or a naive CD8 positive T cell with its T cell receptor will bind to MHC1 and its accessory receptor, which we'll draw like that. There's CD8. So the T cell receptor and CD8 will bind to the MHC antigen complex, right? And at the same time, if there's a helper T cell, see now we'll say this isn't just a naive CD4 positive T cell anymore. This is a helper T. So a helper T and a CD8 positive T cell will bind to that antigen presenting cell at the same time. And the helper T cell, when it binds to MHC2 with the antigen via its T cell receptor and its CD4 receptor, you may remember back when we were talking about B cell activation, what happens when a helper T cell binds to MHC2 and the antigen and recognizes that antigen as non-self. Go back and look, right? The same thing happens here. It will release interleukins. So it's going to release interleukins, and the combination of those interleukins and this interaction here will cause the CD8 positive T cell to differentiate into a cytotoxic T cell, or T subscript C. And I guess I should clarify here. Like that. Okay. So now this CD8 positive T cell has been activated. It's a cytotoxic T cell. And what these guys do is they're killers. An activated cytotoxic T cell will kill any cell expressing that antigen on MHC1, except for antigen-presenting cells. So now that this cytotoxic T cell has been activated, this cytotoxic T cell can make a bunch of copies of itself, and then they go out into the body and they just start hunting for that antigen on MHC1. And if they encounter that antigen on MHC1, what it means is that that cell is making a protein that it shouldn't make. And so the cytotoxic T cell will kill the cell that's making that protein. I realize this may be kind of confusing, I strongly encourage you to read the book when it comes to T cell activation, because it can be kind of confusing. This is a kind of a complicated dance that happens, and it's a little bit more complicated than what I put on here. This is somewhat simplified, but use your book. Use the office hours. Come to office hours, and we'll talk about this. I'll help you with it as long as it takes for it to click for you. And use each other. You know, you've got Flipgrid. Use Flipgrid to really help each other understand this stuff, because I think that, you know, teaching this kind of material is the best way to really understand it. Okay, so yeah, so that's T cell activation. So now that we've got all these activated cytotoxic T cells that are going to go out and kill any cell expressing that antigen, let's talk about how they actually do it. So they have what we call T cell or cytotoxic T cell killer proteins. They work a whole lot like the complement system. So there's two types of proteins here. There are perforins and granzymes. And so the way this works, let's say here's the target cell that's being targeted by a cytotoxic T cell. The perforins, you know, they look a whole lot like complement. The perforins create a hole in the membrane. They perforate the membrane. That's why they're called perforins. Yeah. And then the granzymes can enter the cell through the perforins and cause apoptosis. And I think on the next slide you can see, yeah, there's perforins right there. If you look on the next slide, the perforins look a whole lot like the complement. And so on the right you can see the perforins in the plasma membrane, and the granzymes are basically just small enzymes that can pass through the perforins and start the process of apoptosis in the targeted cell. Right? So this is how T cells will kill. This is how cytotoxic T cells will kill any cell expressing MHC2 with the targeted antigen. So one strategy, one possible strategy that viruses could use, right, to get around this would be to switch off the expression of MHC2, right? That makes sense, you know, because viruses, they can switch on and they can switch off different genes. So one possibility is that a virus could get into a cell and switch off the expression of MHC2. Right? And then essentially that cell would be flying under the radar, because if a virus switches off MHC2 in its host cell, then the host cell is no longer expressing those viral antigens on MHC2, and it's not, you know, making itself a target for those cytotoxic T cells. But luckily we have cells called natural killer cells. And natural killer cells, call them NK. This is another type of lymphocyte. And they have no T cell receptor, right? Because their job is basically to identify and kill any cell not expressing MHC2. So if the virus happens to switch off MHC2, then the natural killer cell will kill whatever cell stopped making MHC2. Or I'm sorry, not MHC2, MHC1. And a good figure to end on is this next one. Yeah. That next figure on the next slide, this kind of basically summarizes all those different elements of the adaptive immune system, because there's two parts to your adaptive immune system. There's the B cells, the antibody part, and then there's the T cells, cytotoxic or helper T cell part, and it's all kind of working together. So take a good long look at that figure. You find it in your book. It's figure 20.20. Yeah, so study that figure, or use that as kind of a framework to study all the things that we've been talking about in this chapter. So one thing that I've mentioned a few times, it's this idea of self and non-self. I've said it a few times that the T cell receptor will bind to an antigen on MHC1 or MHC2. And then if the T cell recognizes that antigen as non-self, then XYZ happens. But that kind of opens up the question, how does a T cell know? How can a T cell determine whether or not an antigen is self or non-self? And so it has to do with the way that they develop, right? The process of T cell maturation and selection. This is something that happens in the thymus, and it's something that happens very early on during the life of an organism that has a thymus. So basically what goes on in the thymus very early on during development is you have all these cells called thymic APCs, thymic antigen presenting cells. And what the thymic antigen presenting cells do is they'll go through the entire genome, right? They'll go through all the genes in their DNA. And they'll just start making every single protein in the entire genome. And just making those proteins just to stick them on MHC1 and MHC2. So these thymic APCs present random endogenous proteins on MHC1 and MHC2. So this kind of makes them unique. They can express endogenous proteins on MHC2. And so the process of T cell maturation is a two-step process. You have T cells that are maturing in the thymus in the presence of these thymic APCs with all of these endogenous antigens on MHC1 and MHC2. And it's a two-step process. Step one is called positive selection. And basically what happens is if a T cell receptor can't bind to the major histocompatibility complex, it's useless, right? What's happening, or what happens in the T cells before they even get to this step, is the gene for the T cell receptor just gets rearranged, and the rearrangement of that variable region is totally random. And sometimes the result is that the T cell receptor can no longer recognize the major histocompatibility complex. Well, if that's the case, then that T cell receptor is totally useless. So positive selection ensures that only T cells that can bind the major histocompatibility complex live. All the other ones get killed. So that's positive selection, right? Negative selection ensures that the T cells that survive don't bind too tightly to these antigens that are being presented by the thymic APCs. So if a T cell, or we'll call it negative selection, if a T cell binds too tightly to the antigen MHC complex on the APC, so if the T cell receptor binds too tightly to the antigen MHC complex on a thymic antigen presenting cell, comma, it is killed. Ooh, that's bad. I'm sorry. It's been a long day. I'm trying to cram like half a semester of lectures into 3 days. Cut me some slack. Okay. So basically, how do we put this? When the T cell receptor is being rearranged, right, before this process of maturation happens, when the T cell receptor is being rearranged, there's basically three possibilities. There are three possible results when the T cell receptor gene gets rearranged. Yeah, we'll write those down over here. Possibility one is that the T cell receptor gets arranged in such a way that it can no longer bind to MHC. The T cell receptor no longer binds to the major histocompatibility complex. All right? That's one possibility. Another possibility is that the T cell receptor gene gets rearranged in such a way that the T cell receptor recognizes a self-antigen. That's another possibility. The third possibility is that the T cell receptor gets rearranged in such a way that it can bind to MHC, but it doesn't recognize self-antigens. So possibility 3 is a T cell receptor binds to MHC, but not any self-antigens. So when you see recognize, another good way to think about this is bind. If you get a nice tight bind between a T cell receptor and an antigen, that's what I mean by recognize. Okay, so these are the only 3 things that can happen after you rearrange that T cell receptor gene. All right. Which one of these do you think is the good one? Right? Well, if a T cell receptor can no longer bind to MHC, it's useless, right? So 1 is bad. We don't want 1 to happen. But thankfully, positive selection ensures that only T cells that can bind to MHC live. All others killed. So because of positive selection, that doesn't happen. What about if the T cell receptor binds to self-antigens? Well, that can cause a lot of problems, right? So, but thankfully, if a T cell receptor binds too tightly to one of these antigen MHC complexes on the thymic APC, it gets killed. So during T cell maturation, if you wind up with a T cell receptor that binds too tightly to a self-antigen, it gets killed. So what this does is it ensures that every T cell that makes it out of the thymus has a T cell receptor that can bind to MHC, but not to any of the self-antigens that were made by these thymic APCs. They've already been exposed to all the possible self-antigens that your body can make in the thymus, and they haven't bound to any of them. But because they passed positive selection, we know that they can bind to MHC. So this is how all the T cells make it out of the thymus, ideally. All right? Ideally, I say that because sometimes a rogue T cell can slip through the cracks and make it out of the thymus with a T cell receptor that can bind to self-antigens. And when that happens, you wind up with something we call autoimmunity, when you are immune to yourself. So this is what happens when the immune system produces antibodies or T cell receptors that can target self-antigens. And so this results in a type of disease called an autoimmune disease. Some examples, some that we've already talked about. Type 1 diabetes. type 1 diabetes mellitus. In this case, the self-antigen that gets targeted is a protein on the beta cells of your pancreas. Another one. Multiple sclerosis. Right? This is what happens when your body produces, or your immune system targets proteins on your oligodendrocytes. Right, that's a cell we were talking about in AMP1, right? It produces, this is what makes a lot of the myelin sheaths in your central nervous system. Rheumatoid arthritis. Rheumatoid arthritis, this one is what happens when the synovial membrane of your joints gets targeted. And so you get a lot of real heavy inflammation in those joints. And the treatments for all of these, for a lot of them, is just immunosuppressants. Right? So the idea is just to suppress the immune system so that it stops attacking itself. So one really good example is corticosteroids, right? Hydrocortisone is, a great treatment for a lot of these, to an extent. You don't want to take that stuff for, too long, but in acute cases of autoimmune diseases, it can do the trick. And that's it for chapter 20.